Today the topic of discussion would be on nucleobases, nucleosides, nucleotides, structure of DNA and DNA replication. Now the nucleobases or the nitrogen bases present in the nucleic acids are purines and pyrimidines. Now purines are adenine and guanine, whereas the pyrimidines are cytosine, uracil and thymine. Now purines are adenine and guanine, and a purine base joins with a ribose molecule or a deoxyribose molecule to form a nucleoside. If a ribose molecule joins with a purine, it is known as a ribonucleoside, whereas if a deoxyribose joins with the nitrogen base, it is known as deoxyribonucleoside. Now the adenine will with ribose will form adenosine, and adenine with deoxyribose will form deoxyadenosine. Now the guanine will form guanosine, and with the ribose moiety, and with the deoxyribose, guanine will form deoxyguanosine. Now, as far as the pyrimidines are concerned, cytosine can form the nucleoside, that is cytidine, which may be either deoxycytidine or cytidine. Now, in the case of uracil, the pentose sugar is always ribose, and hence the nucleoside form is uridine. In the case of thymine, the pentose sugar attached to the thymine is almost always deoxyribose, so it is known as thymidine. Now adenine, guanine, thymine and cytosine are the bases, nitrogen bases present in deoxyribonucleic acid, and whereas the adenine, guanine, cytosine and uracil are present in the ribonucleic acids. Deoxyribose is present in the DNA, and ribose is present in the RNA. Now the nucleosides, if they are attached to phosphate, they become the nucleotides. So nucleotide consists of a nitrogen base, a pentose sugar and a phosphoric acid molecule. So from adenosine we get adenosine monophosphate or AMP. From guanosine we get guanosine monophosphate or GMP, and from uridine we get UMP, that is uridine monophosphate. From CMP it is cytidine monophosphate, and from TMP, from thymidine it is TMP, that is thymidine monophosphate. Now these monophosphates of these nitrogen nucleosides, or rather nucleosides, can be converted to the diphosphates and further to the triphosphates. So we have ATP, GTP, CTP, TTP and UTP. So they are the triphosphates. Now in the triphosphates, the last two phosphate bonds are high energy bonds. For example, if the ATP becomes ADP plus PI, then it liberates 7.4 kilocalories of energy. Or stated otherwise, if we want to convert ADP plus PI to ATP, then we need 7.4 kilocalories of energy. Similarly, the other nucleotide phosphates also. Now, there is a cyclic nucleotide. There are cyclic nucleotides, for example, cyclic AMP and cyclic GMP. Now, the cyclic structure is obtained when the 3-5- phosphodiester is bound as far as the ribose moiety is concerned. Now, the numbering of the atoms in the purine or pyrimidine ring and that of the pentose sugar are different. So, for the formation of cyclic nucleotide, there should be a phosphodiester bond between 3-carbon and the 5-carbon of the ribose or deoxyribose moiety. So, cyclic AMP it can be produced and cyclic GMP also can be produced. Now, in the body, ATP is acted upon by adenyl cyclase. Now what is formed is cyclic 3-5-AMP, and if the guanylate cyclase acts on GTP, what we get is cyclic 3-5-GMP. Now cyclic AMP and cyclic GMP are intracellular signals of various extracellular events, including the binding of or the action of hormones. That means they are the second messengers. And mostly the function of cyclic AMP and cyclic GMP are found to be in opposition to each other. So now the cyclic AMP and cyclic GMP are destroyed by a phosphodiesterase enzyme, and hence the cyclic AMP and GMP cyclic GMP are converted to ordinary CMP and ordinary GMP. Now this phosphodiesterase is inhibited by caffeine, which is present in tea and coffee, and so excessive drinking of tea and coffee may lead to excess amount of caffeine, and this may inhibit the destruction of cyclic AMP and may cause excessive lipolysis, and hence is not conducive for good health. Now, after finishing these nucleotides, we will see something about the structure of deoxyribonucleic acid. Now, deoxyribonucleic acid is a double helical stranded structure in which one strand is running from 3' to 5' and the opposite strand runs from the 5' to 3'. That means the two strands are anti-parallel in nature. Now the bonding between the adjacent nucleotides on the same strand is 3', 5' phosphodiester bridges, whereas on the opposite strand, that means the bonding of the nucleobases of the opposite strand is by hydrogen bonds. That is the binding or the pairing is between A to T, which has got 3 hydrogen bonds, and the other pair is the guanine and cytosine, which has got 2 hydrogen bonds. So A to T pairs with 3 hydrogen bonds and G to C pairs with 2 hydrogen bonds. This is known as the base pairing rule or the Chargaff's rule. This results in the number of purines and pyrimidines are almost equal. That means A is the concentration of A is always equal to thymine, and the concentration of guanosine is always equal to cytosine. Now, as it is a double helical structure, it can be compared to a spiraling staircase, with the footsteps can be compared to the hydrogen bonds, and the railings are the 3', 5' phosphodiester bonds. It can also be considered to be two parallel roads, each carrying traffic in the opposite directions. As it is a helical structure, it also results in the formation of grooves There are two grooves, one minor groove and the major groove, and to these grooves the various proteins can be interacting and thereby regulating the expression of the genes. Now, and it also makes the DNA to exist as nucleoproteins. Now, there are two types of proteins seen, histones and non-histones. Now, histones are basic proteins which are rich in lysine and arginine. There are 5 types of histone, H1, H2A, H2B, H3 and H4. So these are the major histones. They are basic proteins, histones and non-histones, they regulate the gene expression. Now histones also, or the proteins, especially the histones, protect the DNA molecule from the deleterious action of deoxyribonucleic enzyme. So it is protected, it forms a protein coat over the deoxyribonucleic acid. Now, as and when the transcription or replication is to take place, then the histones may have to be detached. Actually, higher organization of deoxyribonucleic acid, if we observe under electron microscope, we can see spherical structures called as nucleosomes, which are connected by DNA filaments. Now the nucleosomes are formed by histone octamer, 2H2A and 2H2B and H3 and H4, and these are forming the nucleosomes, and these nucleosomes are interconnected by the H1 histone and also the DNA filaments. Now this is a nucleosome structure which is most predominant in the mammalian DNA, whereas in the prokaryotic DNA the nucleosomes are not present. The nucleosomes may be about 20 nucleosomes or so. They twist around each other to form DNA filaments, and then further they form into chromatin fibers, and then chromatin fibers further coiled to form the chromosomes. Now we have 23 pairs of chromosomes present. Now next we will talk something about the DNA replication. Now DNA replication takes place with the help of several enzymes and proteins, and DNA replication takes place only once in the lifespan of a cell. So a particular cell is divided only once, and so the DNA replication also takes place once in the lifespan of the cell. Now there are two different phases in the cell cycle. That is, there is a synthetic phase, there is a mitotic phase, and there is a G1, G2. And G1 and G2 phases where the proteins are formed, and it is in the synthetic phase where the DNA replication takes place, and in the mitotic phase the cell division occurs. So the entry of the cell from one phase to the other is controlled by special proteins known as cyclins and some other enzymes like cyclin dependent protein kinases. So these two are very important in the regulation of the cell cycle. If the cell cycle, if the cyclin proteins appear in large quantities or at improper times, that may affect the cell division and may cause a cancerous state. So the synthetic phase requires the uncoiling of the supercoil of the DNA. And actually this DNA exists as a negative supercoil which is almost folded 8,000 times. So this has to be uncoiled first, and this is helped by the topoisomerases 1 and 2. Now in the bacteria there is bacterial gyrases. Now mammalian topoisomerases, they are helicases, and they uncoil the DNA. Then there are another set of enzymes known as helicases, which actually separate the two strands of DNA by hydrolyzing the hydrogen bonds between the two strands. Now, in this manner, replication bubbles are formed, and one of the strands of DNA is synthesized in the 5' to 3' direction, and it is known as the leading strand, and the other DNA strand is replicated again from 5' to 3' direction only, but this is done in the opposite direction. And it is also it is done in small fragments. Now this strand is known as the lagging strand, and the strand which is replicated continuously is known as the leading strand. Now lagging strand is replicated as Okazaki pieces, where there is a small RNA primer of about 20 nucleotides, ribonucleotides, and the 21st nucleotide onwards it will be deoxyribonucleic. So the various enzymes like topoisomerases, helicases, and DNA polymerase are required for the DNA replication. Now, to keep the two strands of the DNA separate during the replication process, also required are the single strand binding proteins which keep away the two strands so that re-annealing will not take place. So in this manner, but the DNA, it is quite somewhat interesting to note that the DNA replication actually starts with a small RNA primer, and you need a primase enzyme also for this. So a small RNA primer of about 20 or 25 nucleotides is formed first at the 5' end of the new strand and the 3' end of the parent strand. And the next nucleotide onwards, it is a deoxyribonucleic, and this is catalyzed by the DNA polymerase enzyme. Now this is what is happening in the leading strand, but in the lagging strand, the DNA is replicated in short fragments known as Okazaki fragments. Now, after several, most of the DNA is replicated, the RNA primers have to be removed, and the gap left after the removal of RNA is filled up by the properly base paired deoxyribonucleotides. Now, the selection of nucleotides to be attached to the growing strand of DNA depends on the base pairing rules. The base pairing rule, as I have stated, is A to T and G to C. So there is very less chance of a mistake taking place. Now the DNA replication can be compared to taking photostat copies of all of the pages of the book, whereas RNA transcription is taking the photostat copies of only selected pages of the book. And we have to exercise great accuracy for the DNA replication, because the daughter cells should be getting equal information, the correct information from the parent DNA molecule. So it's a it should be a very accurate process, and this base pairing rule takes care of this this accuracy, there should be a lot of there are several control points. It is something like a double monitoring system. Now, after the replication is over, certain other enzymes they further screen the sequences, and if there is any mistake that is occurring, it is cleared, it is corrected for this purpose. So fidelity, this is known as fidelity. Fidelity means great accuracy and loyalty is required in DNA replication. Now there are so many damages that can take place to the DNA, and these damages may be due to UV rays or X-rays or gamma rays, and there may be one base changes or a portion of the DNA can be deleted, and one example is thymine dimer formation, which may be taking place due to the UV rays, and this has to be corrected by thymine dimer cleaving enzyme. Now if we are not able to do this correction, then we suffer from genetic disease known as xeroderma pigmentosum, where there is a presence of skin cancers. So like this, there are several types of DNA damages occurring, and if we are not able to repair these DNA damages, then it might result in cancerous states. For the DNA repair mechanism, there is nucleotide excision repair, base excision repair also, missense matching repair. So if these mechanisms are not functioning, we get several types of diseases. So the basic mechanism is to find out where the mistake is lying, and cut the DNA near to that point, and certain DNA polymerases find out the correct base and insert the correct base at the gap, so that the correction of DNA has taken place. Now we can say that those organisms which are able to survive today, they have all the mechanisms to correct the DNA damages, and other organisms which are not able to correct the DNA damage are not able to survive today. Now last word about the telomere portion. Now telomere is the end portions of the chromosome, and due to the continuous replication process, the end portions of the chromosome, that is the telomere, gets shortened, because this part cannot be replicated. So there is a shortening of the chromosomes. So a 20-year-old man's DNA and a 70-year-old man's DNA, if you compare, then there is a shortening of the 70-year-old person's DNA. Now, in most cases, there is an enzyme known as telomerase, which will restore the length of the chromosomes. In this manner, telomerase helps to restore the length of the DNA polymerase. In cancer cells, it is found that telomerase enzyme is more active, and a way of treating cancer can be to inhibit the telomerase enzyme, and research is progressing in this direction.